Alright, so now I'd like to talk about glucose metabolism. Okay, in the previous unit we looked at the immediate system and kind of how that worked. You know, it's the first system to kick in. So now I want to talk about the next system to kick in. That's your anaerobic or uh, glycolytic system. Okay, so glucose metabolism, we're going to look at that. We're going to look at a few other things relating to glucose and how it's metabolized, both stored and broken down. Okay, so it's going to be kind of an overview of all things glucose at the metabolic level. Okay, so first off, let's start with a little bit of nomenclature. Okay, as we go through, I know we're going to start getting into a realm of big nasty words, for lack of a better term. Okay, so it helps you if you can kind of just look at a word and kind of maybe figure out, based on some of the parts of it, what it's trying to say. So let's look at this. All right. If I look at this glycolysis, okay, glycolysis, you've seen it before. I mean, in high school in biology classes, I'm sure you go over this. Glycolysis, simple. Anything that has the first part of the word of glyco refers to glucose. Okay, so glyco, and then we have lysis back here. Lysis is a term for breaking something down. Okay, so glycolysis. This is the breakdown of glucose. Okay, pretty simple. We'll talk about it a little bit. There's a bit of a misnomer within glycolysis. Some people will say the term an anaerobic versus aerobic glycolysis, and it's not correct to say because glycolysis is always anaerobic, but the things that come from it, the end product of that process, can be either metabolized aerobically or anaerobically. So that's kind of where the misnomer comes from. But just know it's not correct to say that. So if you're in those in a circle of scientists or something, you can be we'll say looked down upon for saying such a thing because it's just not technically correct. Okay, on to the next one. Let's look at this. So we have glycogenesis. Okay, glycogenesis. So same word, glyco. So glucose, genesis, like the Bible, right? So creation of something. Alright, so we have creation of glucose. So there's a few processes where we have this occurring, and we'll look at that later. And then we have here glycogen, glycogen, we'll just, hold on, just cut that one out. Glycogen, a lysis. Okay, so glycogen is the stored form of glucose, which we'll talk about how that kind of is created later on, but glycogen is the stored form of glucose in the body, and then we have that lysis term again. So this is the breakdown of glycogen. And as we'll see, glycogen is broken down into this form of glucose, which is glucose 1-phosphate. Alright, and that's it. Once again, it seems like a huge term and whatnot, and if you're not familiar with it, that's fine. I'm going to talk about it, and you're going to see it's actually pretty simple. Alright, just keep an open mind as we go. Alright, we're going to be throwing a lot of terminology around, and I just want to make sure you guys are up to pace with everything. Alright, so let's move our epic pen here. Alright, so once again, as I said, glycolysis is anaerobic. By definition, it does not require oxygen at any step of the process of breaking down glucose. We don't need oxygen, so it is truly an anaerobic process. Now, where the misnomer kind of comes in and how people will sometimes say aerobic versus anaerobic glycolysis is the end product of glycolysis is this molecule called pyruvate, okay, or pyruvic acid. All right, so pyruvate or pyruvic acid, the only difference between those two things, if you see these two different terms, they really refer to the same thing. Okay, pyruvate is the same thing as pyruvic acid. The difference is pyruvic acid has an extra hydrogen attached to it, and that's it. Okay, so one's the oxidized, one's the reduced form, but they're the same thing. Okay, so if you ever see pyruvic acid versus pyruvate, for our purposes, it's the same thing. Okay, So, pyruvate is produced from glycolysis. Okay, it's the end kind of, if you will, it's the reason you're even doing glycolysis. Okay, so you're trying to get these pyruvates, and they can be kind of routed two different ways. One of which being going to lactate, or as you mostly have probably heard of this, lactic acid. Okay, so you can have lactic acid being produced, or you can have this thing called acetyl-CoA, which we'll talk a lot more about as we go on. But that acetyl-CoA molecule is used for aerobic metabolism, whereas lactic acid is not. So that's where the misnomer comes in, is what people are trying to say when they say that is, um, at the end of glycolysis, does the pyruvate go toward an anaerobic destiny, if you will, or fate, or does it is it being converted into something that's going to be used for aerobic metabolism? Okay, and that's where the difference is. But once again, the actual glycolytic or glycolysis uh, reaction is always anaerobic. Just the end product is sometimes routed different ways. All right. Now, something to be aware of as we go along. We're going to start talking about how we're getting ATPs and numbers of ATP and all that kind of things, and kind of tracking how much energy we're getting from molecules of things. Uh, Glycolysis, as we'll see in the next slide, is a fairly lengthy process, and for the most part, we're getting our glucose is going to be coming in right at the top, as we'll see, but it can also come in from the side, and that would be through glycogen breakdown. And glycogen, once again, the stored form, nets us 3 ATP, whereas glucose gives us 2 ATP. All right, so there's a bit of an advantage to breaking down glycogen, and we'll see as we go a bit more about that. But I just wanted to preface all that. Okay, so here we go. This is that equation. All right, so this is glycolysis. Up here we have glucose, and let's talk about the different places, right? Blood glucose, that's coming, as you can imagine, from your blood being absorbed in to the muscle, okay? And then it's getting broken down into all these intermediaries, okay? And these different intermediaries are, really just shuffling around the parts of that molecule, adding things, subtracting things, getting split in half, as you see here. It starts with 6 carbon, and then down here it splits into 2 3 carbon molecules, okay, and then it goes all the way down, and we get pyruvates from it. Okay, so we get, if we look at the net equation, 1 glucose comes in, 2 phosphates, 2 ADP, and 2 NAD. And we're going to talk about NAD a little bit later. And then from it, we're getting 2 pyruvates, 2 ATP, 2 NADH, 2 free hydrogens, and 2 water molecules. So we are creating water here. We're getting more free hydrogens, okay, which, if we're thinking about a whole body acidosis thing, right? We're talking about how much acidity is in the body. Acidity is just how many hydrogen molecules are freely floating around, give or take. Okay, so if we're producing hydrogen here, as we saw with ATP hydrolysis as well, we're contributing to a drop in pH and acidosis of the body. Not really for this lecture or this unit even. We're going to talk a lot more about that in the cardiovascular unit, but thought I'd throw it out there. So let's look at this reaction a little bit. Okay. So as you're looking at this, you might be noticing the net reaction doesn't perfectly line up with the actual reaction. Okay, so right here, the first thing, when we take this glucose and we want to start processing it, it costs us an ATP, so we're -1 right now. Okay, now we go to this step down here, and look, we just used another ATP just to get this going. So now, before we're getting any energy out, we're down -2 from this part right here. Okay. So once we go down far enough, we're going to take these NADs, stick a hydrogen on them, and they're going to either go to this way or down here. We'll talk about both of those later. Um, but then as it splits in half, we start generating ATP. So right here, we're plus 1, plus 1, plus 1, plus 1. Okay, so from this part right here, we can see we've generated plus 4. Okay, so the reason I write it as a net equation, it's that, you know, we are generating 4 ATP, but it's costing us 1. Oh, sorry, it's costing us 2 in order to get it working, so therefore, as a net of those two things, we're actually only generating 2 ATP from breaking down a glucose molecule. Now, as I mentioned on the previous slide, if we're getting that energy, if we're getting that glucose molecule from a stored glycogen, we actually get a lot more energy from it, and here's why. Because if you look at it, if we're getting energy from muscle glycogen, we skip this first step. Okay, it just comes in right to the side, and whammo, we get to go right here, which means we're not losing that 1 ATP. Okay, which then means that we're only at -1 here, and the net of these two things, right, would be +3 instead of +2. Okay, so we get a little more energy when we take energy from glycogen. All right, and as I said before, the kind of the end stage thing we're getting, these pyruvates. It's all about the pyruvate in glycolysis. All right, pyruvates are either going to go to lactic acid or lactate, or they're going to go to acetyl-CoA, and they're going to go into this Krebs cycle, and we all know how fun that is, so we'll talk about that later, but that's the aerobic process that the pyruvate can go in. So those are kind of the fates, if you will, of pyruvate. There's more things that they can go into, but from a bioenergetics perspective, these are really the two things that you'll talk about. Okay, so let's talk about glucose a little bit. I'm not going to require you to memorize this and draw out the chemical structure or anything, so don't get all PTSD on me or anything. I'm just the reason I put this up here is that as we look at the different terms and you're looking at that huge 12-step process of glycolysis, you may be seeing terms like let's go back here a second. We have a glucose 6-phosphate, glucose 1-phosphate, fructose 6-phosphate, okay, fructose 1,6-bisphosphate, okay. Kind of to let you understand what that means. That's why I put this molecule up, all right, because the numbers involved in those terms typically just refer to where things are attached to the various carbons on the glucose molecule, all right, and that's really about it. So that's fine. We're just gonna leave that as is. But this is what a glucose looks like from a or D-glucose, which is the form of that's the form of glucose in humans anyway. So D-glucose. There's different ways this molecule can kind of be arranged, but this is what it looks like inside of us. All right. Okay, I'm sorry this is a little bit blurry, but it's a good depiction. So let's talk about the fate of pyruvate a little bit. All right. So as I said, glucose it gets broken down. We get pyruvates. We get a little bit of ATP. Honestly, netting 2 or even 3 ATP from that long multi-step process isn't giving you a whole lot. Okay, realistically, because if we looked at something like the immediate system with the phosphation system, where it's a single step process and we're getting 1 ATP from it, it's a pretty nice fast clip as far as turnover goes. Okay, glucose breakdown takes a little while longer, and the reason is you get a better product at the end of it that we can use for other things. So anyway, pyruvate, as I said, is either going to go toward lactate or acetyl-CoA and produce a little bit of CO2. All right. Now, the two enzymes which act upon pyruvate are lactate dehydrogenase or pyruvate dehydrogenase. So if you ever see this on a test or something, and you see lactate dehydrogenase, it probably means that it's turning it into lactate. Okay? And the other one, just by process of elimination, would be pyruvate being acted upon by pyruvate dehydrogenase would form acetyl-CoA. Okay? So, to a certain extent, the nomenclature helps you there a little bit. Now, let's talk about why this happens to a certain extent in a second. But so if in the case of these two different things, if we have lactate production, it's going to build up in the muscle a little bit. The muscle will try to process it in-house, and then eventually it'll spill out into the blood. At that point, that's when when you're exercising, that's when you're starting to feel the burn. Okay? So that's what that is. When your muscles start to burn a little bit, that's just lactic acid being produced, and you're starting to feel that. That's one of the few tangible metabolic processes you can actually kind of feel easily. So otherwise, acetyl-CoA will be shuttled over to, well, actually it ends up being in the mitochondria. Okay, so glycolysis is occurring out of the mitochondria. It's occurring in the sarcoplasm or the cytoplasm cells. Okay, so it's closer to the muscle fibers, kind of like the immediate system is. Just takes a bit longer to do, but same general area. Acetyl-CoA needs to be processed in the mitochondria, so that's a bit of a different step, and we'll talk about that. And then also we're getting CO2 production, and that is relevant to the pulmonary system and the cardiovascular system. And we'll talk about that in a different unit. Alright, so let's talk about lactate a little bit. Okay, lactate gets a lot of bad press, if you will. Whoever the PR guy was for lactate, he did a bad job. So lactic acid. To this day, there's a lot of people out there who blame lactic acid for just about everything. From muscle damage, soreness, to fatigue, pretty much anything that happens in the body, lactic acid has probably been blamed for it at some point or another. And a big reason for that is humans are really bad, untrained humans, I'll say, okay? Because the human mind needs to be trained to a certain extent. Untrained humans are really bad at making correlations, okay? And the reason that lactic acid gets blamed for so much stuff in exercise is because it's pretty much the only thing you can feel during exercise, metabolically, okay? So, you start producing lactic acid about the time when you start to fatigue from high intensity exercise. Now, it's not the lactic acid that's causing you to fatigue, but you're producing a ton of lactic acid because the metabolic systems are starting to fatigue. So, as the systems are becoming fatigued, you're feeling lactic acid production, you're feeling that burn, and then a lot of people make the relationship in their mind and they say, oh, well, the lactic acid, the burn, is causing me to fatigue. When in fact the two things just happen to coincide at the same time. They're kind of just happening at the same time. It's not one causing the other. Okay, so lactic acid. Let's talk a little bit about that. Lactic acid, just like I said before with pyruvic acid, the only difference between lactic acid and lactate is a hydrogen. Lactic acid has an extra hydrogen. Lactate is missing that hydrogen. All right, and that's pretty much all there is to it. They're the same thing for our purposes. Okay. Now, once again, these brackets refer to concentration. So lactic acid concentration. So the concentration of lactic acid in your blood is related essentially to intensity of work or intensity of exercise. Okay, and that's related to homeostatic disturbance, and that is how much of your musculature are you moving at any given point. Okay, you don't have a ton of lactic acid hit your system if you're just, say, like walking at a leisurely pace down the street. Most people are going to be fairly okay to do that without getting a burn. Um, but if you really disturb this system, do some sort of like a hit training, or you know, take a dare and do 100 burpees as fast as you can, you're you're going to have a lot of disturbance within your body. You're going to be using a lot of energy at a very fast pace. You're going to be using a lot of glycogen. All this stuff is going to cause you to have more lactic acid in your system. You'll become more acidotic. All right, not just from the lactic acid produced from the ATP hydrolysis, from the different other metabolic systems producing hydrogens, but essentially at the end of the day, um, you're going to get more lactic acid in your system. You're going to become um, more acidic a little bit. You have systems to recuperate that, but anyway, not this lecture. We'll talk about that later. So why do we even produce lactic acid? You know, there's some things in the body that really just don't make sense. Like, you know, we could talk about autoimmune disorders. Some of them we don't can't put our finger on. Lactic acid isn't one of those. Lactic acid actually has a very relevant and important job within the metabolic system, okay, within the bioenergetic system, and it has everything to do with this molecule NAD. All right, and what happens when lactic acid goes from NADH to when lactic acid is produced, pyruvate is catalyzed by this enzyme, lactate dehydrogenase, and part of that reaction of taking this pyruvate molecule and turning it into this, okay, is donating a hydrogen, taking this hydrogen off of NADH and putting it on there. Yep. And that leaves us with an NAD that's missing that hydrogen. And this seems probably pretty relevant for you right now. And we're going to talk about that in a second, okay? But this is really why this reaction even exists. It's the purpose for it. Okay? Oop. So, NAD. NAD is a molecule called nicotinamide adenine dinucleotide. For the sake of my not having to say that too much, I'm just going to call it NAD. Okay? And it's used, we're going to see it a bunch, honestly, throughout this bioenergetic unit. It's used in a bunch of reactions, both in the mitochondria and you will see it in glycolysis as well. Okay? And this molecule is used to grab up hydrogens, essentially. There's various reactions within glycolysis that cannot proceed if the hydrogen remains on the substrate, or the thing that's being acted upon to produce a product. Okay, so the NAD serves to take hydrogens off of things, and in doing so, it becomes reduced. Okay, so not to delve too much into chemistry or anything here, but there's this term oxidized versus reduced. Okay, and I put a little box over here for you guys. But essentially, the oxidized version of something is when it's missing a hydrogen or missing an electron. Okay, and the reduced form is when it's got it. Okay, so if it's oxidized, it means it's ready to take up a hydrogen, and if it's reduced, it means that it's already got one. All right, and these guys kind of help to make metabolic systems progress and keep going. They have a lot more relevance later down the road, but for our purposes, we're just going to talk about how they help the glycolytic reaction right now. And this is pretty much it. Okay. If we look at this big equation of glycolysis, okay, once we get down to these steps right here, okay, right here, and then yeah, just right here, okay. In order for us to take this molecule right here and turn it into this one, we have to um, put a hydrogen on this NAD. Okay, so you see here, it's NAD plus. It's ready to take a hydrogen. It's going to take a hydrogen off of this guy, and that helps to get this reaction going. Okay, if we can't do this process, if this gets knocked out, then we can't do this. Okay, and instead of our glucose turning into pyruvate, our glucose turns into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is not useful for us. We need pyruvate. At this point, if we were unable to do this reaction right here, this NAD goes to NADH, and glyceraldehyde 3-phosphate turns into 1,3-bisphosphoglycerate. Well, if we can't do that, what we've done is we've taken glucose, we've invested 2 ATP, and nothing's come of it. Okay, I'm not much of an investor, but if I'm putting resources into something and I'm not getting any kind of result from it, I'm going to go bankrupt pretty soon. It's not good. So, can't have that. Metabolically, it's not favorable. So, here's our problem. Our problem is, as we start doing glycolysis, we start doing this. Okay, we start taking the concentration of NAD. Okay, we start taking this concentration versus concentration of NADH. Okay, and this is in the sarcoplasm. Okay, so we're in the sarcoplasm here. Okay, and within the sarcoplasm we have a certain amount of NAD. Okay, it's a cell and it's kind of hard to think about, but basically what you have is a test tube, if you will, and it's got a certain amount of molecules of NAD. All right, let's just make the math easy on us. Okay, so we got 100 of them. All right, and we got 0 of these. Now, as we start doing glycolysis, now we're gonna maybe slash this down to 80, and as this reaction right here starts to progress, well, now we have, say, 20. Okay, and we're still able to keep doing this because we still have NADs available, right? But eventually what can happen, theoretically, is if we couldn't turn these NADHs back into NAD, we might run out of NAD and have a bunch of NADH. Now, NADH is useful in a different energetic system, but for glycolysis in the sarcoplasm, it's useless. I get nothing out of that. If I can't do this, I don't get this. I don't get this, and I don't get this or that. I get nothing. Okay? Don't get this energy, don't get that energy. I get nothing. Okay? So, it's very important that I'm able to kind of recycle these things, right? Because as I'm taking this NAD and I'm turning it into NADH, I need some means of turning that NADH back into NAD, recycling it back, so that this glycolysis reaction can keep moving. All right? So, enter lactic acid. The process of creating lactate converts NADH back to NAD. So, as you might expect, when I'm doing glycolysis very fast, when I'm breaking down glucose at a very fast rate, and I'm starting to flood my sarcoplasm with NADH, I will start to do this more often, and I'm going to start to have more lactic acid production. Okay, versus, and there's another way we can do this as well. NADH can be recycled through something called electron transport, and that's something we'll actually talk about in a little while. It's a much slower process. Okay, within metabolics, everything comes down to location, substrate availability, and speed. Okay, so if I need energy really, really fast, I can't rely on a slow process. This is a single step right here. This step, instead of me going here, if I go here, I can regenerate this, okay, bring it back over to here, and I can keep letting this process go, all right? And uh, I can keep recycling, and I can allow glycolysis to keep flowing, all right? I can keep that concentration of NADH low, keep the concentration of NADH higher, and that allows glycolysis to keep moving. Because otherwise, it could stop. And if it stopped, that's true fatigue. You're unable to produce energy. That's problematic, right? So, if we relate this back to, think about real world. When do you actually produce lactic acid? Once again, it's typically not when you're leisurely walking somewhere. When your energy demand is low, you don't typically produce lactic acid. When you're doing something that's of higher intensity, you're doing, say, a 400, an 800 meter run, 1 mile, okay, lifting weights, anything like that, anything it's requiring you to produce a lot of force in a short period of time, is meaning that you're having more cross bridges attached in it per given unit of time. More cross bridges being attached means you're burning more energy, you have a higher ATP demand. If ATP demand is high, you need metabolic systems that are going to produce fast, okay, on site. Glycolysis happens in the sarcoplasm, just like the immediate system. Okay, it's closer to the fibers that are actually using the ATP, so it's a preferable energy system. It's not as fast as the immediate system, and it takes a while to get going compared to the immediate system, but it still will get you ATP production. Okay, that's why it's the second major system that kicks in along the way toward getting to oxidative metabolism. Okay, so it's not the fastest, as you can see. There's a bunch of steps here, right? But it's still on site. You don't have to cross membranes. It's easy enough to get substrate or glucose to burn. Okay, it's a pretty fast system. It's not the fastest. You don't get the most energy out of it, but it's pretty fast. So, that in a nutshell is why you produce lactic acid. If you're working fast, you're more likely to produce lactic acid. If you're working lower intensity, something that doesn't require you to produce a lot of force very fast, you have more likelihood of not producing lactic acid and going toward Krebs cycle. And you end up getting more energy that way, but that's skipping ahead. So we're gonna talk about that in a little bit. All right, so let's talk about how we recycle that lactic acid. Okay, so as I said, we produce lactic acid, it builds up in the muscle. Eventually, you'll see it spill out into the blood, typically. Okay, and that's the point of lactate threshold. Okay, but we don't, it's not a waste product. Some people like to talk about lactate as being some sort of a useless waste product. It's actually just an intermediary, okay? Just like a lot of the other things we talked about, it's just an intermediary. So if we look here, the Cori cycle is part of the gluconeogenesis chain, which we'll talk about as we go along. And what happens is, if our muscle here, say we produce lactate, okay, and lactate goes out into the blood. Same thing with pyruvate. Pyruvate can leave the muscle and go out into the blood if needed. Okay, so these guys together, when they hit the liver, either as lactic acid, lactate, pyruvate, okay, they can be reconverted back to glucose or even stored as glycogen. This is one of the ways that your body tries to maintain glucose in the blood, because this blood glucose is exceptionally important to maintain in order for you to have healthy living and performance. Okay, so that's a little bit in the future. We'll talk about that, but just know it's really, really, really important that you have enough blood glucose for your survival. So as we go along here. Gluconeogenesis. Okay, this is a mouthful, I know, but if we break it down once again, glucose, new genesis. So creation of new glucose. Okay? Gluconeogenesis. It's a big word, not really that frightening once you look at all the parts of it. Okay, so your body has a lot of redundancies for making sure that its blood glucose levels are pretty homeostatically held. Okay, if they get too high, you damage tissue. If they get too low, you die. So, in a nutshell. Anyway, we can create new glucose from a lot of different sources. You can break down muscle, create amino acids. Amino acids can then be converted into either acetyl-CoA or pyruvic acid. Pyruvic acid can then go up and be turned into glucose. We can take acetyl-CoA, move it up the chain, create glucose. We can take triglycerides. From triglycerides, we can get glycerol or fatty acids. Glycerol can then be turned into glucose. Fatty acids can be turned into acetyl-CoA. Okay, you see where I'm going with this. So, we have a lot of different things. Essentially, we can in a roundabout way take just about anything you can eat and turn it into glucose. By the way, when we're talking about something like post-exercise nutrition and stopping catabolism, if you happen to be low on blood sugar, this is the process that's going to take your muscles, tear them apart, and get glucose out of them. Okay? Like a loan shark. So to speak. The shakedown. Anyway, if your body wants glucose, just give it sugar. You don't have to give it protein, so the protein gets turned into amino acids, then pyruvic acid, and then eventually goes to glucose, right? Instead, if you just give it sugar, this process stops and you preserve your protein, your muscles. You're precious. Okay, so you can't read this right here, so I'll read it out. Fast muscles have a greater gluconeogenic capacity than slower muscles. Okay, so for the most part, gluconeogenesis occurs in the liver. Some of it does occur in skeletal muscle, and it is not energy saving. It requires as many as 4 ATP just to make the glucose. And as we saw from the process of breaking down glucose, you only get 2 glucose out of it. That's just how important glucose is to the body. It'll lose energy to make sure that it can create the glucose molecule. Alright? So that's the process of gluconeogenesis in a nutshell. We'll talk about this more as we go. Okay, so lactate threshold. Lactate threshold can essentially be defined as the point where your muscles are unable to process lactate in-house, so to speak, inside the muscle at a great enough rate to compensate for how much is being produced. At a certain point, because even right now, as you're sitting down watching this video, you're not working very hard, I'm presuming, from a metabolic standpoint. You are producing some lactic acid. Okay? There's always pyruvates being broken down, and they'll be acted upon by lactate dehydrogenase or pyruvate dehydrogenase. It's just the ratio of which one's acting on it has a lot to do with intensity, but you're always kind of producing both. Okay? So, but you're probably also not feeling a burn right now because that little bit of lactic acid being produced in the muscle is able to be processed in-house. Now, as you start working and you start going to higher intensities of work, you start producing more and more lactic acid. Lactic acid, at a certain point, will overwhelm your muscle's capacity, or whatever muscle it's being produced in, that muscle's capacity to process it and turn it back to glucose. And at that point, when it starts building up to a great enough level, it then starts to spill out into the blood. And that's the point where we call lactate threshold. Okay. From the observation of it, you would just take 10 testing of a blood sample, usually like a finger prick, and you can measure lactate levels within that. And once it accumulates above a resting level, it can be said that you've reached lactate threshold. And it's often used as an indication of training status within endurance sports. Because those who have a higher VO2 max typically have a higher lactate threshold. As you see here, an untrained person will have a lactate threshold, or an LT. Okay, you want to sling around a little acronym here, you can sound like you're in the know. Okay, if you have an LT somewhere around 50 or 60, you're probably untrained. If you're 50 to 60% of your VO2 max, that is. If you're an elite athlete or a higher, more trained elite athlete, your LT will be somewhere around 70 or 80%. Okay. It's not necessarily the case that you're going to be a better athlete in that case. It's just often seen. Right. All right. Now, modality to a certain extent affects lactate threshold as well. And in this case, it makes a lot of sense. Okay. So with biking, you typically see people have an onset of blood lactate accumulation, or their LT is much lower than people who do treadmill tests. Okay. And that's a big difference, right? And this is significant. The reason being, simply, is that when you're cycling, you're only using primarily your quads. I mean, not only, but like that's the major muscle group you're going to use. And when you start to overwork one muscle group, that one muscle group will start to have a lot of energy demand. In that case, it starts producing a lot of lactic acid. You'll start seeing blood lactate levels accumulate faster with cyclists, simply because they're not able to pull as much of their body into the motion. Whereas if you compare that with running uses the majority of the muscles in your body, just each of them to a fairly low degree, unless you're doing something like hill climbing. That's a whole different thing. But if you're running on a flat track, you're using your arm swing, you're using your core to stabilize, you're using, of course, your quads, you're using your calves, you're using a lot of your body to accomplish that motion. And that being that you're able to distribute the work across more musculature, each of the individual muscles don't have to work as hard, which means they don't produce as much lactic acid. It's kind of the nuts and bolts of that. All right. So, if we look at a graph, okay, here we have lactic acid or lactate is the purple line, and the dotted blue line here is heart rate. If we were to plot, down here we have wattage, okay, wattage of work versus blood lactate here on the left side, and then heart rate is on the same axis but on the right side of it, okay, and then up at the top here we have VO2 max. Okay, if we plot all these things, OBLA is onset of blood lactate accumulation, right? So this is kind of what lactate accumulation looks like. If you ever really do test somebody, which I hope to do, honestly, well, we can't do lactate, but we can do blood glucose. Anyway, if you test somebody for blood lactate through exercise and you take them near max or to max, what you'll see is a fairly steady or constant increase in heart rate. Okay, heart rate doesn't typically do this. Okay, it doesn't have that kind of exponential increase. Okay, instead, it kind of just progressively goes up with workload. Okay, so as intensity goes up, it goes up as well. Blood lactate is going to stay low, stay low, stay low, and then as it starts to creep up over about this 2 measurement here, okay, then you hit lactate threshold, and then you have this exponential increase, okay, curvilinear increase, and that's typically what you see. And this also kind of corresponds with fatigue response. People, as they start to work harder, once they start having a good amount of lactic acid in their system, they're also starting to generally reach point of fatigue as well. Once again, lactic acid is not the thing causing the fatigue. It's just when you're working really hard, when you're having a lot of metabolic activity occurring, you're also producing a lot of hydrogen, secondary to many metabolic systems, and that's one of the major things that will cause people to fatigue, is the accumulation of hydrogen ions. But there's any number of other reasons we can pin fatigue to. Anyway, this is kind of the progression of things. So let's talk about how we create glycogen. All right. So glycogen is, if you were to ask me what is the single most important factor governing an endurance event, a soccer match, a cross-country race, basketball, football, most sports, if you ask me what is the single most important thing that will tell me whether or not somebody is going to perform better than somebody else, okay, whether we're going to have a good performance, it's glycogen level. The amount of blood sugar the amount of glucose stored in the muscle, in the liver, is probably going to be the most important factor governing performance. All right. So, glycogen is stored in the body. It's essentially excess glucose. Your body says, "Hey, this stuff's really important. I should keep some of it on hand." Okay. And depending on training status, you can have more or less of this molecule. Okay. Glycogen is a polysaccharide. It's a very, very, very big glucose molecule. Okay. So, anyway, when you start running out of glycogen, as I've said, it's super important. When you start running out of glycogen, you fatigue. Okay. Simply put, when your glycogen drops off, you stop moving. At least, you stop moving fast. So, there's a term, hitting the wall. It's often used in endurance sports. Some of you may be familiar with this. But when you hit the wall, it means that you have... people often talk about, when they hit the wall, feeling as though they're moving really fast. But to any outside observer, their speed has moved to a crawl. And the reason being is at that point, you have depleted to a great extent your body's glycogen level, and you're starting now to, from a performance standpoint, the problem is you're starting to try to draw on fats as your primary energy source. And while great for weight loss, it takes a lot of time to get energy out of that, and if you can't get energy fast, you can't move fast. So that's why people's performance drops off. The second, more negative thing to all this is your body needs that glucose. So if you've burned it all up running a marathon, now people's bodies are literally telling them, stop moving, we could die. Because you really, really, really need glucose. It's the only thing the brain will run on. And if your brain stops getting energy, it stops working. I don't really need to explain why that's bad. So, hitting the wall. I thought I would include a quick little video of a pretty iconic Ironman race that was performed a while back. So, anyway, I'm going to cut to video real quick. It's only a few minutes long. It's a good watch. I would recommend it. And it just kind of underscores and gives you an image, so to speak, of what hitting the wall really looks like. So let's look at that real quick, and then I'll pick up back with the lecture.
Shawn Welch is in a desperate spot. Her body has run out of fuel. No calories to burn. Systems shutting down. The legs are there, you just can't feel them. The eyes still see, but through a gauzy veil of delirium. At this point, Sean Welch is aware of two things. The finish line is so close, and someone is so close behind. After more than 140 miles, it's come down to less than 100 feet to that damn line. Just yards behind, her stalker appears. It's Wendy Ingram. Their bodies were obliterated, but the spirit held firm. Welch and Ingram crawling in the dramatic spirit of Julie Moss. In a battle for fourth place.
Okay, so I hope you guys enjoyed that video. It's a good watch, like I say. Very enlightening. It's good to kind of put a face to some of the things we talk about. All right, and that was the instance of these two ladies giving it all and really moving to a point of true metabolic fatigue, and that's being wiped out of glycogen. So this is how we create glycogen. All right. As I said, glycogen is kind of a really big carbohydrate molecule. It's a polysaccharide, okay, and it starts with glycogenin. Okay, that's kind of like a protein that's kind of at the core of glycogen, and if you have excess glucose, right? If you say you exercise or something, you take down something sugary afterwards, and now your body's kind of has more glucose than it needs, well, then it's going to start to store it. Okay, and what it does is it takes these glucose molecules and it binds it to glycogenin, and it just starts making a chain. Okay, so it makes this chain out this way. Okay, and at a certain point, when the chain gets long enough, then it starts branching out. Okay, so you have one chain here and another one branching out like that. And the end result of all this, as it gets big enough, is eventually you start having these really complex structures. Okay? And this is our glycogenin molecule in the center here, this protein. And from there we have one strand which is multi-branched out here, and each one of these is branched out, and you can see there it's kind of like a snowflake at a certain point, right? And that's kind of what glycogen looks like. Now the benefit to this is, as opposed to just having, say, one really long strand like this, the way that we break down glycogen is kind of the opposite of how we put it together. It's one linkage at a time. Okay, so if all these were glucose molecules, then I would just have to do one cut at a time. And if I had one chain, that would be a really slow process. Instead, I get more surface area. So if I need to break down glycogen, I can act on that one, that one, that one, that one, that one, that one, and so on and so forth. So I can react with the whole outer shell, if so to speak, and keep wearing this thing down all at the same time. And I can break down glycogen very fast if I need to. Okay? And that's kind of why this molecule is the way it is. Now, one of the problems with storing glycogen is one, it requires water, and two, its spherical shape doesn't really make it conducive to being tightly packed. Okay? If you look at it, it's kind of like a bunch of popcorn in a bucket, right? Popcorn. Okay? You can jam it into a bucket, but you're not getting tightly packed. You're going to get little air pockets, right? You're going to have spaces in between those molecules, and to a certain extent, that's why you can't store a ton of glycogen in the body, as opposed to fat. Fat actually does. It folds and makes itself very neat as it stores, which means you can store an awful lot of it. All right, so let's look at the breakdown of glycogen, or glycogenolysis. Okay, so as you would expect, essentially what we have is the opposite of before. Okay, so we're going to break down these side chains. Once we do, we break a linkage, okay, here. All right, and then eventually you break down the overall chain, and that's how we do it. Okay, we're not too worried about the different enzymes within the process. That I included them here, if you're so inclined to look at that. But essentially, you just start breaking down and releasing individual glucose molecules, so that your muscle, say, can take it up and do that glycolysis process. All right. So, glycogen's entry into glycogen entry into glycolysis. Okay, as I said, as I showed you before, we actually end up saving one glucose, so we get a little more bang for our buck, so to speak, if we go from muscle glycogen. And that's just simply changing around this molecule a little bit. Okay, because we start with glucose, and glucose has to be converted through being catalyzed by hexokinase, glucose 6-phosphate. All right. So instead, we go from muscle glycogen, and a phosphorylase enzyme will get us to glucose 1-phosphate. So what does that mean? I want to give you a quick illustration of what that means. All right. That's all this is. Okay. So if we have glucose 1-phosphate, if you remember back, this is the 1 position of the glucose. Okay. And this is the 6th position of the glucose. 6 carbons in a glucose. So so we have the same number of things, okay? All of our hydrogens, oxygens, carbons. Each one of these is a carbon, by the way. Okay. All right. Okay. So we have the same number of carbons here, because there's a carbon there. This is the 1 prime, 2 prime, 3, 4, 5, 6. Okay. The only difference, these two molecules, the only thing that's changed is that enzyme, that phosphorylase, has taken this phosphate and taken it from the 1 prime position and moved it to the 6 prime position. So it's the same stuff, essentially. It's just being moved around, and that slight change in the structure of that molecule means that it can enter here and then start going down the process. Okay. Now, direct relevance, you guys have been going like, why are you showing me this? I just want you to understand that a lot of the biochemical things that are happening in your body all really come down to minor things, like changing orientation of a phosphate in this instance. That makes something either able to go through a metabolic process or useless. Okay, so it's important and also realize that it's not as intimidating as you might think. Okay, so now let's talk about the oxidative cycle, or the aerobic system. Alright, so oxidation, the aerobic system. The aerobic system operates within the mitochondria. Alright, so the mitochondria, with that, it's going to have a mitochondrial membrane. Remembering back, we have a membrane around the cell. Now we got a membrane around the mitochondria. So creates a bit of a problem because everything, if I want to get energy out of the mitochondria, I have to get stuff into the mitochondria to be broken down. So I have to cross a membrane. And as I said, within any kind of bioenergetic reaction, within metabolic reactions, if I'm trying to get ATP fast, the more borders I have to cross, the slower it's going to be, right? Because often you have to have, things actively transported across. It's just not as easy to do as opposed to something like just, hey, the reaction is right here, right next to the place that needs the energy, so I can do it really fast, okay? So that's one of the reasons the aerobic system is a bit slower. You have to get stuff into the mitochondria, okay? And often, and all the reactions are long, and they're slow. Now, the upshot of all this is you get a lot of energy. By comparison, just tons of energy as compared to the glycolytic system. So this is the citric acid cycle. You can call it the TCA cycle. You can call it Krebs. It's all the same stuff. It just depends on which textbook you use and who you talk to. But the citric acid cycle, named after citrate. Okay, that's all that is. Krebs or TCA, doesn't really matter. All right. So, let's talk about this a little bit. So, acetyl-CoA. Acetyl-CoA is something you're going to have to get used to hearing about a bit. Okay, acetyl-CoA is the main thing coming into this. If glucose was the substrate for glycolysis, acetyl-CoA is the substrate for the TCA cycle. All right? And it can be formed by a number of things. Like I said, pyruvate can form it. We can go from fatty acids. We can go from amino acids. Okay, there's a number of ways you can create acetyl-CoA, which is why the aerobic system can burn anything, right? Amino acids are protein. Fatty acids are fat. Pyruvate comes from carbohydrates. So those are, if we talk about macronutrients, that's protein, carbs, and fat. You can burn anything within this system. Anything that you can eat, as long as it's a macro. I'm not talking about some pica person here, but as long as it's a macronutrient, it can be burned in the aerobic system. So that's a real benefit to this. So once this acetyl-CoA comes into the cycle, it forms with this oxaloacetate molecule, and it forms citrate. Okay, and that's the first step. Once again, why you're getting this name here. All right. Now, through this process, you're getting a bunch of intermediaries. This is kind of like a metabolic master key reaction. Okay. Now, we're going to talk about it from a bioenergetic perspective, but realistically, you get parts for DNA, you get parts for cellular regulation, we get parts for building muscle. We have a lot of intermediaries for a lot of different processes being produced within this cycle. Okay, so just understand what we're talking about is just a very small part of why this is relevant to the body. All right, so let's look at this. What are we getting in? What are we getting out of it? So first off, like I say, we're putting in acetyl-CoA in. All right. From that, we're going to derive CO2, CO2. Okay, we have two CO2s coming out. All right. So when you're breathing out carbon dioxide, for the most part, that CO2 is coming from this cycle. There's a few other places we can get it, like the conversion of pyruvate to acetyl-CoA will give you a carbon dioxide, but the majority of it is coming from the Krebs cycle. So that's where the CO2 that you're breathing out is coming from. All right. And now we're also going to start deriving NADHs. NADH, NADH, and NADH. So we're getting 3 NADHs. We're getting something called a GTP. It's a guanine triphosphate. For our purposes, it's an ATP. Energetically, it's about the same. So you're getting like 1 ATP, and we're going to get an FADH. Okay? So, FADH, one of those, 3 NADHs, some carbon dioxide, and an ATP. And we have our net reaction down here, which I've written out. Okay, so acetyl-CoA plus 3 NAD, 1 FAD. Those are the main things going into it. So we need these guys in order for this process to occur. And we're going to get NADH, FADH, more hydrogens, ATP, and some CO2. That's about what we're getting from this. And the other system is the electron transport system, ETS. If we're looking back at the other one, right? Let's go back here. We have all these steps occurring, and we want energy. We get one ATP, one from this whole thing. It's a lot of steps to get 1 ATP. This, a bit like glycolysis, is upfront investment, which gives us more yield on the back end. Okay, this is we're putting this into a mutual fund or something. Okay, we're investing for the long haul. Okay, so electron transport system is that pay dirt. It's the place where we're getting like almost all of the energy that you're using on a day-to-day basis. Okay, massive amounts of ATP, and we're deriving that ATP directly from those NADH molecules and FADH molecules, which we're producing throughout the way. Okay, so there's actually 2 different reactions here, whether it's NADH or FADH. Okay, so if it's NADH, say that's where we're getting our energy from, NADH is going to go into the electron transport chain, okay, and it's called the electron transport system for good reason. The system actually just moves electrons around, okay, and through moving those electrons around, we don't need to talk too much about the different protein structures, but the net result of all of it is that we can take ADP, okay, adenosine diphosphate, and we can directly jam phosphates back onto it to create ATP. This process is the place where you're using oxygen in your body, for the most part. Energetically, it's the only place. So when we take a deep breath and we breathe in that oxygen, this is the system that we're trying to feed. This is the place where we're going to use that oxygen. All right? That underscores to a certain extent, or it should, just how important this system is, right? Because how important is it breathing oxygen? You know, we've seen multiple systems that were anaerobic and allowed us to not use oxygen. But if you're without oxygen for several minutes, you're dead, right? If tissue doesn't get oxygen for several minutes, it dies. Because you're not able to produce enough ATP through those other systems to keep your tissue alive. This is your big energy system. Alright, so from this, we're going to produce a little bit of water. We're going to use oxygen. We're going to use the hydrogens. Of the different systems we've seen, they pretty much all produce hydrogen. This actually uses the hydrogen. Also, within Krebs, we're producing NADH and FADH. This, also located within the mitochondria, is using those things, okay? Using that hydrogen, okay? And the ADP and phosphate, we're going to get those from ATP hydrolysis occurring within the muscle. Okay, so at the end of it, once we've taken this ADP and put a phosphate back on top of it, we have our ATP, which we can then go and use. We got a little bit of water, and we got two NADs. And these two NADs will then just go back, and they'll allow us to keep working on that Krebs cycle. Kind of the same way that lactate dehydrogenase process allowed us to have glycolysis keep working. All right. So, well, before we go any further, I want to mention something here. It's a bit interesting. You guys have maybe heard that vitamin B is an energy supplement, and if you haven't, it's a thing. The supplement industry, if you look at it from a historical perspective, it just goes in cycles. So something goes in, a few years later, people realize it really doesn't do much. Manufacturers of that stop selling it. 10 years later, once everybody's forgotten that the thing didn't work, they put it back on the shelf again, and they talk about how amazing it is. You'll see. You'll live long enough, you'll see. That's kind of how the way things work. But anyway, vitamin B being sold as an energy supplement is because of this reaction and glycolysis and Krebs cycle. Okay, reason being is NADH, that nicotinamide adenine dinucleotide, is a direct result of, I believe it's vitamin B2 niacin. I might be off, might be B3, but niacin. Niacin is actually converted into NAD in the body, and FAD is or flavin adenine dinucleotide. Okay, so is part of riboflavin, another B vitamin. Back in the 1920s and prior to that, before people started to enrich wheat and grain products, people weren't getting enough vitamins, so they would actually have vitamin B deficiency. And if you didn't have enough niacin or riboflavin in your diet, then your levels of NAD and FAD were actually low. And if those levels are low, then the electron transport system doesn't have enough substrate to work. Krebs moves really slow because it doesn't have its cofactors here, these NADs and FADs, right? So they can't really function too well. And then glycolysis, still it needs NAD, right? So if you don't have those vitamins, then yeah, those metabolic systems go slow. If your metabolic systems are going slow, then you by definition are lacking energy. So like in the 1920s, if somebody had a bad diet and they were impoverished and they weren't having enough vitamin B in their life, they could go to a doctor and get a vitamin B shot, and like within minutes of introducing vitamin B back into their system, they immediately perk up. They would immediately get energy. And that's why if you ever watch these old black and white Wile E. Coyote cartoons, they'll take a vitamin B shot and they'll inject it, and then they, get all wild, presumably because they have more energy. Anyway, while that is a supplement, and it is useful if somebody is deficient in vitamin B, very few people this day and age are deficient in vitamin B, because nearly every processed food that you eat is fortified with vitamin B for this very reason. So if you're not deficient in it, taking mega doses are not actually going to help you at all. It's just going to end up being eliminated next time you go to the bathroom, because it's water soluble. So that's why vitamin B is said to be an energy supplement, because 100 years ago it was. Alright, so let's move on a little bit. So hydrogen shuttles. Hydrogen shuttles are another means of getting hydrogen off of NAD in the sarcoplasm to help glycolysis work, but also it allows you to take the hydrogens that were on those NAD and use them for energy. Okay, so you have two different hydrogen shuttles. You have the glyceryl phosphate shuttle, and you have the malate aspartate shuttle. And the difference between the two is just which which place they're located. Okay, so in type 2 muscles or fast twitch muscles, you use the first one. Okay, so this is going to be type 2. This is type 1. All right, type 1. And so what they do is a hydrogen shuttle actually shuttles the hydrogen off of an NAD, and it moves it across the mitochondrial membrane. All right. In the case of a glyceryl phosphate shuttle or a type 2 hydrogen shuttle, type 2 muscle fiber hydrogen shuttle, it goes from NAD here, NADH, okay, it's taking the NADH, and it's going to take that hydrogen and put it onto FAD, okay? So you're going from NAD on this side to FAD on this side. Okay. Now, in a type 1 shuttle, it goes from NAD to NAD. All right. That's about all you need to know there. So NAD to NAD. Now, the relevance of this. If you go back and you look at the equations for the electron transport system, which we saw a second ago, NADs are worth approximately 2.5 ATP per. FADs are worth approximately 1.5 ATP per. So, in a type 2 muscle fiber, if we're using those hydrogen shuttles, we're actually losing 1 ATP. Reason being, well, this is per hydrogen being shuttled, reason being is we're going from NAD to FAD, right? So, if NAD is worth, NADH I should say, is equal to 2.5, and FADH is equal to 1.5, as a net, we're losing 1, okay? And this one's just equal. We're not losing anything because it's going NAD to NAD. In the glyceryl phosphate or the type 2, you're actually losing a bit of energy, okay? And that's one of the reasons, one of the many reasons why a type 2 muscle fiber is less energy efficient when compared to a type 1. Alright, let's move on. Alright, let's talk about fatty acid utilization now. Okay, so we've talked about how we can take glucose and tear it down. We've talked about the immediate system. Alright, so let's talk about fatty acids real quick. Alright, so fats are the hardest thing in your body to break down. Okay, that's why once you put weight on, it's a lot harder to take it off than it is to put it on. A lot less fun too, typically. So here's some of the problems we have. Okay, fats or lipids, as we're often called, are stored toward the outside of the body. Okay, they're stored in adipose cells. Okay, we can actually call fat, we can call it adipose tissue as well. Whatever. Okay, it's roughly the same. All right, so it's stored toward the outside of the body, which means that the place that we're storing the energy, the fuel, the fat, is definitely not close to the place we're burning it. Okay, so it means that if we want to break it down, we have to go out there and get it from outside of the like toward the outside of the body, toward the skin, break it down out of those adipose cells, like jailbreak it, so to speak, get it into the blood, take it into the blood. Now it has to be transported into the muscle, but we can't use it in the sarcoplasm, so we need to now transport it into the mitochondria, and at that point we can start to break it down, which is a whole nother step on top of Krebs and on top of electron transport system. So what we have is just a lot of barriers, a lot of things in our way before we're able to use fat as a great energy source. Just because there's a lot of transport involved. It's bad logistics, all right? So that's kind of how it works. So the first thing we have to do is perform something called lipolysis. Lipolysis, once again, looking at the term, lipo, lysis, okay? Fat breakdown. Okay, we're going to break down lipids. Okay, lipid breakdown. And lipolysis looks something like this. We take a triglyceride. So we have a triglyceride. Triglycerides are made up of glycerol, glycerol, and 3 fatty acids. Okay, so 3 fatty acids. Okay, that's a triglyceride, right? So that's how we store fat in the body. In your adipose cells, you have 3 fatty acids, and they get attached to a glycerol head. Okay. Now, if we want to break down and get that fat out of the body, or sorry, out of those adipose cells and get them up to use them, we have to rip them off that glycerol head, and then get free fatty acids. Okay. Free fatty acids can then enter the bloodstream. Glycerol enters the bloodstream as well. If you remember back, glycerol is used in gluconeogenesis, so we can create glucose out of that. And then these guys, these 3 fatty acids, are going to be taken up and be broken down. Alright, so once we do that, once we have the breakdown of fat, we have lipolysis occurring. It has to go out to the blood. It has to bind up with an albumin, a protein carrier, so to speak. Okay, so that's going to grab it. And then once it makes its circulation run through your circulatory system, and it finds a muscle that wants to take and grab it up, it now has to be carried through. Okay, so we have to actively transport that fat through. So great, we get our fat, we bring it in. Like I said, now we need to get it into the mitochondria. And this is where something called carnitine comes in. You guys ever heard, perhaps, some of you have, of a supplement called L-carnitine? L-carnitine, the L in front of it just refers to the isoform or the type. Okay, the purpose of that supplement is to increase your ability to transport free fatty acids into the mitochondria. Okay, because that is the carrier protein. Okay, once you get fats into a muscle cell, you now have to get that fat into the mitochondria. Fats bind with carnitine and are pulled into the mitochondria. So that's the idea of that supplement. Hate to be the bearer of bad news. It doesn't really work. If theoretically you could actually directly inject it into every one of your mitochondria, maybe it would have an effect. Problem is, your body doesn't have any issue with producing carnitine in general, so you're never really deficient at it, or deficient in that protein. And if you're not deficient, a supplement doesn't really do anything. So, it's a bit of a waste of money. So, anyway, back to the topic. So, we got our fatty acid, we broke it out of its adipose cell, we broke up that triglyceride, it went through the circulatory system, it got into the muscle, through the sarcolemma, and now it's going to traverse the sarcoplasm, get into the mitochondria, get transported through. Now we have our one fat molecule into the mitochondria. Now we have to start doing something called beta oxidation. Beta oxidation is the process by which we take that fatty acid chain and we start breaking it down into acetyl-CoA molecules. Okay, so and once again, so once we get acetyl-CoA out of it, well then we can just use it in the Krebs cycle, and at that point everything's the same. All right. So if we do look at this though, Fatty acids are just made up of a bunch of carbons chained together. Okay, that's why we call them long chain molecules. Okay, they're just chains of carbons. So acetyl-CoA is a 2-carbon molecule, and if we say we have, in this case, we got what, 2, 4, 6, 8, 10, 12. So this would be a 12-carbon fatty acid. All that beta oxidation does is it cuts that chain into smaller bits. It cuts that chain into 2-carbon acetyl-CoA molecules. And in doing so, every time it performs one of these cuts, or one of these cleavages as it said, we derive one FADH, and we also get one NADH. Okay? So, what is that? Yeah, right here. So we get one NADH, one FADH per cleavage. Okay? And if we remember our energetics here, NADH is worth 2.5, FADH is worth 1.5. Net result, we're getting 4 ATP every time we do a cut. Because this is all occurring in the mitochondria, so we can use these NADHs and FADHs in the electron transport chain. And then we're getting energy from each of these acetyl-CoAs. So this, you can see it's a very dense energy. Alright? Fatty acids give you a ton of energy once you can actually break them down. It's just really hard to get them to the point of being broken down by comparison to other molecules. But if we look at the energy yield of this, okay, in this case we're going to take 18 carbon stearic acid, or it's a very common fatty acid of the body. Alright, if we did that, look, we have 1, 2, 3, 4, 5, 6, 7, 8 cleavages, okay, which gives us right here 8 NADHs, 8 FADHs. That combined, because 8 times 4 yields 32 ATP. Now, if we have 9 acetyl-CoAs, which you get here, 1, 2, 3, 4, 5, 6, 7, 8, and 9, okay. Once we get our 9 acetyl-CoA here, each turn of the Krebs cycle yields about 10 ATP, all said and done, between 3 NADHs, 1 FADH, and the ATP. You're getting 10 ATP. Okay, so now we have 9 acetyl-CoAs. They all enter the Krebs cycle, so we have 9 times 10. Then we get 90 ATP. So at the end of all this, we are getting 122 ATP. Now, it costs 2 to transport it into the cell, so subtract 2. Net result is 120. 120 ATP from one fat molecule. Let's think back to that glucose molecule. Okay, by comparison, if we have one glucose that goes, say, the anaerobic route at the end, if it goes to lactic acid, we get a grand total of 2 ATP out of that. Okay? We're getting 120 out of this fat. It's a ton of energy. It's just, like I say, really hard to get to. This is why in high-intensity work, you're never going to burn fat. You just can't break it down fast enough. You can't get it out of your adipose cells, get it into the bloodstream, get it across membranes, do beta oxidation, do Krebs cycle, do electron transport system. You can't do all that in a short period of time, which means you're just not going to be able to do that during a high-intensity work bout. So whenever you're doing a HIIT training or some sort of a very high-intense work, you're not burning fat. You may burn fat afterwards during your recovery period, but during the work, you're burning carbohydrate and you're working with your immediate system. You're not going to burn fat. So let's actually look at the energy yield from a glycolysis, from a glucose, okay, or glycolysis. Now, as I said, if glucose goes and we get our 2 ATP, and then we're working really fast, so we need to keep glycolysis going, and by extension we have to release lactic acid. We stop here. Okay, you get 2 ATP. There's nothing else. But if that pyruvate that's derived from the glucose goes to acetyl-CoA instead, and it gets brought into the mitochondria, well, each glucose molecule gives us 2 turns of the Krebs cycle. And as we remember, you get about 20 ATP. That gives you about 20 ATP because you get about 10 per turn there. The NADHs that are being produced from glycolysis itself, depending on the type of muscle fiber, which is why you have different numbers here. Okay, depending on those numbers, we're going to get something between 3 and 5 ATP from that. We're getting 2 ATP from the actual glucose. Okay, and we're getting at the end of the day, we're getting about 30 ATP. Okay, so if we're getting about 30 ATP from one glucose molecule, it's not bad, but it has to go through the anaerobic route. That pyruvate needs to go and be converted into acetyl-CoA instead of lactic acid. This is going to be occurring at lower intensity rates. If you ever taking an MCAT, for example, or some sort of a quiz, the generally accepted term, although there are differences, say the energy came from glycogen, for example, or it's a type 1 fiber as opposed to a type 2, we can certainly talk about differences in ATP yield, but the generally accepted term is that 30 is the correct answer. You get about 30 ATP from a glucose. Alright, so let's just wrap this up talking a little bit about protein as a fuel source. Alright, so protein, we haven't talked about it a lot as far as an actual fuel source, and honestly we're not going to. Reason being, as it's a bit more convoluted than glucose or fats. And here's why. Okay, if we look at this Krebs cycle here, and we look at our amino acids, all these are amino acids, okay? Leucine, lysine, phenylalanine, tyrosine, all these guys, okay? All these different amino acids, they all enter the Krebs cycle at different points. Okay, so we see here alanine, glycine, styrene, serine, they all go to pyruvate. But asparagine and aspartate, they go to oxaloacetate. And if it's entering it at different stages, you're going to get different energy yields out of it, right? Because if it enters as oxaloacetate, then you're skipping all of this stuff along the way. Or if it's coming in as succinyl-CoA, you're skipping that stuff and you're only getting this much. So instead of doing all that, I prefer to just leave it at, you can break it down and you get ammonia out of it. Okay, those are the 2 things I want you to kind of take out of amino acid production or breakdown. If your body needs fuel, it will break down protein. It won't even bat an eye. It'll break down any protein it can find. Okay? It'll break down the protein in your bones that holds the matrix together. It'll break down the muscle tissue, which you've spent years and years developing. It'll break down any protein. All right. So, in general, it's best to try to keep your body away from burning protein as a fuel, because in general, the protein that we have in our body is not something we want to break down. We want to, eat carbohydrates and let it burn that. Eat fats, let it burn that. Save the protein. All right. But if it were to be broken down, the first step in that process is to remove the amino group, okay, and deaminate it. So when you remove the amino group from amino acids, it's deaminated, then essentially you produce pneumonia. Okay, NH3. So there's two places we've talked about where you get ammonia. One being in a type 2 muscle fiber, trying to keep AMP levels low, so working at high intensities, and also we're producing ammonia through breaking down protein as a fuel, which also typically happens during high intensity work. So generally, if you're producing ammonia in your body, it means you're probably working really hard. And the last little bit here, all right, um, glucose. Okay, so glucose. There's a saying. There's a saying that fat burns in the presence of carbohydrate. Okay, so what that means is that essentially, no matter how hard you want to just burn fat, and maybe cut out glucose so you can burn fat, you really can't effectively burn fat very well without some carbohydrates in your system. And the reason is that in order for the initial step of breaking down acetyl-CoA to function, you need oxaloacetate, OAA, to be in abundance. Okay, so you can either get that through breaking down protein, or you can have glucose go to pyruvate, which then can go to oxaloacetate. And that's the more preferred route. So to a certain extent, if you want to break down fats, you need this oxaloacetate molecule, which is typically derived from glucose. Okay, so if you don't have carbohydrate, if you don't have glucose, you have a hard time breaking down fat, and it can lead to some problems, ketosis being one. But that's one of the caveats of breaking down fat. You kind of have to have carbohydrate in your system.